Hello, everyone. This is Dr. Kellen Cavagnero, and welcome back to another episode of Inflammatory Content, the podcast about immunology and biomedical research more broadly. I'm here to interview world-class researchers to broadly communicate the cutting-edge work going on in their laboratories and to get to know the people behind the science. So sit back, relax, and let's dive into some science. And if you enjoyed the show, please consider sharing the episode, leaving a review, or donating to support my work. Thanks for listening. Today, our guest is Dr. Dequina Nicholas. Dr. Nicholas is an assistant professor at the University of California Irvine in the Department of Molecular Biology and Biochemistry. Her laboratory studies endocrine diseases, including type 2 diabetes and polycystic ovary syndrome. Specifically, her group investigates the intersection of the nutrient environment, the immune system, and metabolic disease using a combination of molecular and cellular biology, transgenic mouse models, cytokine profiling, and flow cytometry. Dr. Nicholas is the recipient of numerous awards, including the NIH Director's New Innovator Award. Dr. Nicholas received her Ph.D. in Biochemistry from Loma Linda University and pursued postdoctoral training with Dr. Barbara Nikolajczyk at Boston University and Drs. Mark Lawson and Pamela Mellon at UC San Diego. In addition to her research, Dr. Nicholas is passionate about changing the culture of science to increase diversity and inclusivity. To this end, she serves as a leader of numerous initiatives, including the UCI KL2 program and First Gen in STEM. Finally, Dr. Nicholas advocates for bringing one's authentic and whole self to science through sharing her ups and downs of being a new PI, a mom, and a minority scholar on social media. So without further ado, please enjoy this conversation with Dr. Dequina Nicholas. So I thought we would start by talking about your research. Your lab works on two different diseases, type 2 diabetes and polycystic ovary syndrome, or PCOS. To the uninitiated, these two diseases may seem very unrelated. How did you come to study type 2 diabetes and PCOS, and how are they related?
Okay, that is actually a really great question. So, most of my training and career, I had no idea what PCOS was. So, I'm going to start off with type 2 diabetes. So, I am a Black female, and type 2 diabetes runs very, very high in my demographic. And in my family personally, my mom has diabetes, my grandma has diabetes on both sides, aunts have diabetes, I'm pre diabetic. And so when I went to grad school, I was like, I got to figure out type 2 diabetes. And that has been like the whole entire goal, like the focus since I've been in grad school, since I've been doing science. I really wanted to solve this. The lab that I joined was actually studying type 1 diabetes. I didn't know there was a difference because no one ever told me there's a type 1 and a type 2. So type 1 is autoimmune, where there's a clear immune mediated attack of the cells that are really important for secreting insulin. That's what regulates your blood sugar. And then in type 2 diabetes, we see this more as multifactorial. If you have a bad diet, maybe some type of genetics may be involved, but either way, over time, your body just doesn't respond to that hormone and it can't use glucose the right way. So you get really high levels of glucose in your blood. And then so in the lab I was in, because they were studying type 1 diabetes, they were an immune lab and trying to understand, why does the immune system start attacking self? Like, it's not supposed to do that. It's supposed to defend us from invading pathogens. And I just found the immune system to be so beautifully complex. It's insane that all these little tiny cells have such big functions. And so I started off there, fell in love with these cells called the dendritic cell. Essentially, they take some peptides or protein or like a foreign molecule, they show it to a T cell and literally tell the T cell, like, hey, this is good, this is bad. This is self, this is non self. And so in grad school, I basically studied that in the context of lipids. Like, how can the immune system recognize lipids? Because lipids are very, very high. When I say lipids, I mean like fats in the body, right? They're really, really high in type 2 diabetes. And a high fat diet, or what we call a Western diet, is a strong inducer or predictor of getting type 2 diabetes. As I went on to my next postdoc, I kept on studying type 2 diabetes, but this time, instead of using like small cell culture models and just looking at the immune cells, I got to get samples from people who actually had the disease and compare to people who were healthy. And what's really funny is that in the middle of that postdoc, I was actually in Boston, and my mentor said she's going to move to Kentucky, and I did not want to go with her. So I was like, I need to find a different lab. And I had a really, really good mentor in grad school who said, look for mentorship as opposed to just the science. And I had met a professor named Dr. Mark Lawson as a grad student who I remember. He came to my poster, he was talking about lipids and thought what I was doing was really, really cool. So I just emailed him. I'm like, hey, do you have a position for a postdoc? He's like, well, not right now, but we can apply to grants together. So we started writing a couple of grants together. I gave him my ideas, he gave me his, and he was interested in how lipids affected a small cell type in the pituitary called the gonadotroph. Gonadotroph basically secretes sex hormones that are important for, like, reproduction. I'm like, "Okay, cool. I don't know much about it, but it sounds interesting, and there's lipids, and I like studying lipids and fats." It had nothing to do with the immune system. When I joined the lab, I realized that I was in a huge department. It's obstetrics, gynecology, and reproductive sciences. Everybody was doing, like, female reproductive health, and this was completely new to me. I had no experience in it at all whatsoever, and in that lab group, I learned about polycystic ovary syndrome. And the more I heard about it, the more they described it, I'm like, "Oh my gosh, this is essentially type 2 diabetes, but of, like, reproduction." And also made me realize that me studying type 2 diabetes, I'm actually an endocrinologist. I study endocrinology. So I decided to merge these 2 fields together by centering it on the cornerstone of what my training was, which was immunology and inflammation. So I basically now look at chronic inflammation, which we know happens in polycystic ovary syndrome. We know it happens in type 2 diabetes, and the whole goal is to, 1, figure out what is causing the inflammation. Is the inflammation upstream of the disease or is it downstream in like a side effect? And then 2, if we understand what's causing the inflammation, it means that we can stop it. And we really want to in the future be able to target the inflammation to help the disease improve. So yeah, that's how I kind of pulled both together. The long story.
I also, going into this conversation, did not know much about PCOS. I knew that it was increasing in prevalence and a reproductive disease syndrome. In my research before this conversation, I found that it's actually similar to type 2 diabetes in that the insulin resistance is a commonality, correct?
Correct, correct. Yeah, when I say it looks very similar to type 2 diabetes, it's very, very similar. So in females, basically, you have this disruption of normal reproductive hormones. So say, for example, luteinizing hormone is important for ovulation and for promoting steroidogenesis, meaning making our estrogen. Like we need estrogen. Everyone does, not even just females, right? And then so this hormone ends up being super, super high instead. So then females don't go through their normal cycle each month. And either they go through fewer cycles or they don't go through cycles at all. And that impacts their overall metabolic health. They have insulin resistance, just like people with type 2 diabetes. And they actually have a very, very high rate of having full blown type 2 diabetes. So you see this concurrent a lot of the time.
Yeah, it's very, very interesting. Going back to the immunological aspects of these two diseases, you said in an interview with Stem Cell that you hope to, quote unquote, determine the antigenic determinants of type 2 diabetes. Are you suggesting that type 2 diabetes is an autoimmune disease like type 1 diabetes?
Okay, I'm going to throw that out there. I am suggesting that it is an autoimmune disease. So this goes all the way back to grad school. And remember I said I learned about a cell called dendritic cell that takes an antigen and shows it to a T cell. And I wanted to know what happens in the presence of fats. I started digging into the literature and I found there is this molecule called CD1A, B, and C. So group 1 CD1 molecules. And these molecules act just like MHC. So MHC is the molecules that take a protein, show it to a T cell. And if you take any immunology class, undergrad, grad school, whatever, you learn that proteins are what make a good antigen. Proteins are can what stimulate the immune system to actually respond and to turn on. And you get taught that lipids or fats are not good antigens. And so we take that as what we call dogma as word, but in all honesty, that's actually not true. In a mouse system, which is what I call most immunologists' favorite tool, because we can do a lot with an animal system. Lipids are not great antigens, but humans have this unique protein, CD1A, B, and C. There is no homolog in mouse whatsoever. So humans have this unique ability to recognize specific lipids and turn on or induce inflammation or an immune response.
And the cells that recognize, that's NKT cells?
No, 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 no. So that's the other misconception that actually happens when I bring this up because there's a molecule called CD1D. So CD1D, mice also have it, and they react with NKT cells. But CD1A, B, and C, they interact with alpha beta T cells and gamma delta T cells. And it is actually a large part of the TCR repertoire. So a lot of T cells can actually recognize CD1A in the human body. They're just not studied because we don't have the models and we don't have the tools to actually do it.
Okay, to clarify, so CD1D also lipid antigens?
Those are glycolipids.
Okay, and so CD1D, glycolipids, NKT cells, mouse and human,
Correct.
CD1A through C, pure lipids, alpha, beta, gamma, delta T cells, only mice.
No, only humans.
Only humans, sorry. Only
Yeah.
See, that's what we need to clarify. Only humans. Okay.
And I could throw it in there too. Pig also.
Pig.
Horsey. hmm So what's been really cool about this finding is that I asked myself the question, like, why does inflammation we see in a mouse on a high-fat diet look different from the inflammation we see on a human or in a human with type 2 diabetes? Why is it that when we try to design immunotherapies from a mouse model on a high-fat diet, they don't work in humans when they cure metabolic disease in mice? And I think the huge discrepancy is the fact that the inflammation is different because we don't have the stimulus, which is the CD1A presenting a lipid and having lipid induced inflammation downstream. It's the reason why you can't take a mouse and solely induce diabetes by giving it a high fat diet. You have to add secondary damage to the pancreas, the place where you would like get the insulin from. The whole idea in my head was, you know what? If I take a mouse and I give it the human molecule, CD1A, can we make the mouse get diabetes like a human just by giving it a high fat diet? And so that's really the project that I founded my lab on and that we started off with first. And it's a big question to approach, and every single step of the way we have to design new tools, which is it's fun. But like the baseline information is there. It's like you give a mouse that has this ability now to recognize lipids through their T cells, they get really bad metabolic disease and they look way more human-like than a wild-type mouse.
I'm curious about the evidence that led you to build this mouse model because that's no small thing to put this CD, 1A into a mouse. There's evidence of autoantibodies against lipids in human patients with type 2 diabetes.
Ha ha ha, that's a great question. So you'll find that a lot of these ideas, they keep going back to grad school, right? So in grad school, I read a paper where someone had seen in rheumatoid arthritis that there were antibodies against palmitic acid. So this is a simple saturated fat, like you can find it in palm oil, you find it in lard. It's very, very prevalent in the diet, especially if the diet is high fat. And I was like, you know what? If you have this high level of antibodies in rheumatoid arthritis, which is autoimmune, autoimmune also has this particular type of inflammation called Th17 that we also see in human diabetes, then maybe we can find the same antibodies in type 2 diabetes, where you know there's usually high lipids. So I collaborated with a school of public health while I was in grad school, and they basically had a biobank of serum from the study called En Balance. So this is a community outreach program where they went to find people who are Hispanic in the community with type 2 diabetes. They use promotores, so basically like community members that they trained to teach people about their own diabetes as an intervention. They gave them glucose meters, gave them interventions on like how to exercise, how often to exercise, like really tangible goals. So for dinner, instead of having like 3 tortillas, you'd have like 2 or 1, like stuff like that. And it was a 3-month intervention, so we had samples from their plasma before, plasma after. We had workups on their A1C before, A1C after. And so with this whole cohort, essentially I have plasma, type 2 diabetes, completely unmanaged, and then type 2 diabetes after a 3-month intervention. And so I decided to build an ELISA where I could detect if there's auto antibodies against these lipids. So what I ended up doing was I biochemically conjugated the palmitic acid to BSA. I coated the ELISA plates with the BSA, so you have palmitic acid kind of facing up, if you can know, like imagine it. And then I would incubate the serum from these patients on the plates. Then add a secondary to detect whether or not I had IgG antibodies or IgM antibodies. And what I found is that in the population with unmanaged type 2 diabetes, you have extremely high levels of these antibodies. But then you see that when you've had the intervention after the 3 months, the amount of antibodies actually decreases, meaning that when you have fulminant type 2 diabetes, it's unmanaged, you have chronic inflammation, that the immune system is turned on and making these antibodies, and that this immune response can resolve. So the fact that I saw the immune response resolving was telling me that whatever was the antigen is there and is present, and that antigen is being cleared, or there's at least less of it. And that's kind of what was the baseline or support leading me towards this hypothesis.
What do you think the antigen presentation of these lipids does to the T cells that leads to potentially the development of type 2 diabetes? What would be the potential pathway?
Okay, so I'm just gonna let you know I'm about to go on a limb here.
Some speculation too is okay, as long as we let people know we're
Cool,
speculative.
cool. So my educated guess would be that when the body presents lipids to T cells, I think the whole idea is to induce a response that helps the body clear the lipids. There's a whole sector of like literature and research showing that too much lipids in the blood is bad for you. It's lipid toxics. Like cells don't like it. They don't live well. They get what we call ER stress and they turn on all these programs that, cause cell death. So we don't want lots of lipids floating around. And I think what we do is have these T cells basically skew into what we call like a Th17 phenotype, and then these Th17 cells, they secrete a cytokine that's very good at inducing class switch recombination to make these antibodies. So then the antibodies would help us to clear the extra lipids that are floating around. But if you keep giving too much of the antigen, which would be the lipid, and it's too high, that means you're turning on the inflammation and you're not being able to turn it off. There's no resolution. And I think it's that chronic activation of the immune system that leads to these cytokines, which normally are supposed to help with class switch recombination. And instead, that's damaging the insulin secreting cells in the pancreas. So that's like the working hypothesis for the disease that we're going off of.
Okay, so do you think that this is happening, this antigen presentation and cytokine response, this is happening in the adipose or in the pancreas or multiple tissues? That's kind of one of the
I
complexes about
agree.
diabetes is like that it's kind of happening in a lot of places. The muscle, right?
Yeah, yeah. So the model that we have, this is the first time we've been able to kind of like look at different tissues and see what might be going on. And the evidence is supporting that most of this interaction is actually happening in the gut. The secondary lymphatics that are in the gut tissue, so like Peyer's patches, the malt, the galt, those would be where we have this interaction actually happening. We have checked like lymph nodes that should be draining systemically, and we don't really see any differences if you put a mouse on a high fat diet and they have this extra like CD1A protein. But you see substantial like increase in size of the lymph tissue within the gut. So we think that's where the interaction is happening. And then what's so cool about that is if you think of all the current hypotheses for what causes type 2 diabetes or what may be important, we know, of course, that like diet is involved, exercise is involved, heredity is involved, like genetics, right? You also have the idea that, the microbiome is involved. And so the fact that we have this immune reaction happening in the gut really pulls together a lot of these disparate or different hypotheses about the origins of type 2 diabetes. It's really quite fascinating.
So when you have this immune response against the lipids, you're getting both a T cell response and then the B cell response that's T cell dependent, right? And you get
Mhm.
the antibodies. Do you think the antibodies and the T cells and their cytokines are both pathogenic potentially, or are you leaning more towards the T cells being the problem and the cytokines, not so much the antibodies, whereas the antibodies are just more of a signature of the autoimmunity?
Yeah, that's exactly how we're thinking about it. Like, I don't think the antibodies are driving disease. I don't think the B cells are the mediators. Part of the reason is that I believe some of this immune reactivity from the B cells can actually be T cell independent. So, in mouse models on a high fat diet, you still get some of these antibodies that seem like they might recognize lipids. And then you can take these like B cells from these mice and try to induce diabetes, and you can't really induce diabetes. So we do think it is T cell dependent. Like all the damage that we think is happening, we think it's coming from T cells and T cells secreted cytokines.
Very cool. Yeah, so now that you have this mouse model, you can test a lot of these things, right?
Exactly.
Use
Yeah.
mice that lack B cells, T cells, and really flesh out how it's all working. That's super cool. Got one more question about type 2 diabetes. I wanna ask you about PCOS again, but one more question about diabetes. So, do you think that the drugs, or do you know whether the drugs that are used to treat type 2 diabetes, any of them like the very popular ones now, like the GLP-1 agonists, do you think they directly influence immune cells?
They may or may not. There is now growing evidence that they can interact with immune cells. But what has me actually very concerned is that if you look at long time course studies, so say you take Ozempic or some other type of GLP-1 like agonist or inhibitor or whatever, if you look at the initial decrease in A1C, so say my A1C is like 7 and I take Ozempic and in like a month it's down by like 2 whole percent points. If you track them over time, it climbs right back up almost to baseline. So whatever we're doing with like Ozempic, we are slowing down the process, but then it speeds back up and we're not stopping progression. So we're not actually targeting what is causing diabetes. So in the short term, it works well. And what we think is happening is that majority of the effects are happening through the weight loss, but we haven't identified or pinpointed what about the weight loss is actually improving type 2 diabetes.
So just to reiterate that, underscore it. So you said Ozempic works well for type 2 diabetes initially, but long term, it's not a good solution.
Yeah, it's kind of crazy.
Now that those drugs are so commonly used for, not only type 2 diabetes, but for just weight loss in general, how they potentially impact the immune system, I think there's a lot of unknowns there that it could be some
Yeah.
PhDs for any people listening. Okay, PCOS. So on your website, you showcase some preliminary findings related to immunity in the pituitary gland, which is not something that I usually think of in terms of immune cells, a place where they hang out. What kind of immune cells do you find in the pituitary, and what role do you think they may be playing in disease and health?
Oh my gosh, this is so exciting. So this has been a pet project that popped up that I am legit falling in love with. So my MD PhD student essentially was looking at the immune cells in the pituitary. And this pituitary, remember, is a really important gland for secreting a whole bunch of hormones that regulate our entire body. And two of those hormones being luteinizing hormone, follicle stimulating hormone, they're necessary for like reproduction. And so she decided to look at what the immune cells were by doing single cell RNA sequencing. Majority of them are macrophages. Like it's a whole bunch of macrophages, which in hindsight makes so much sense. Like every tissue has their own kind of special macrophages that, help the tissue remain healthy. So we said, like, what could potentially be the actual role of these macrophages here? Like, what is the role in this tissue? So she did a very simple, but I think a very thoughtful experiment where she took pituitaries, dissociated them, put them in culture, and basically depleted them of macrophages to see what would happen to hormone secretion. If you get rid of the macrophages, you lose hormone secretion from the hormone secreting cells. So these macrophages are helping to regulate how these cells secrete hormone. She's now been able to actually do this in vivo, which is freaking crazy. And so what's really interesting is, we got scooped on the project, which was a little bit sad at first, but tells us we're not crazy, right? But these macrophages, they really do regulate hormone. And what she's been able to show thus far is that they have very, very specific roles that are different from any other macrophage in the body. So she's compared using Tabula miris, which is basically Chan Zuckerberg has this whole consortium where they like took a whole bunch of different tissues across different ages and just did single cell RNA sequencing. And when she compared, she found that the macrophages in the pituitary express transcripts for growth hormone and prolactin. Macrophages are not supposed to make growth hormone. There's a particular cell type that's supposed to make growth hormone and it's not macrophages. And only macrophages in the pituitary have these transcripts. So now she's on a quest to really prove that these cells are making hormone themselves. And we believe that the macrophages are part of this small network within the pituitary that helps to regulate the overall tissue.
I went in thinking that this is going to be mostly about PCOS, but this is really about all hormone production, which
Yeah.
there's so many hormones produced by the pituitary gland, right? So
Mhm.
that's very interesting. I'm excited to hear, what you all find out there.
Yeah, me too.
Let's now shift to your career path, which has led you to your current role as an assistant professor at UC Irvine. You studied chemistry in undergrad and switched to biology for your PhD at Loma Linda. There, you completed your thesis in the lab of Marino de Leon.
Mhm.
What led to your decision to switch from chemistry to biology?
Well, doing chemistry. Okay, that was that was terrible. Okay, it was very clear that I didn't really like chemistry after I did an entire degree in it. Um, I wasn't bad at it. I just realized, like, okay, this really isn't my thing. It's not what I love. I really enjoyed problem solving. And the whole reason why I chose chemistry in the first place was because originally I wanted to do forensic science, and doing like chemistry, biochemistry like made sense at the
You
time.
watched a lot of CSI as a kid.
Oh my gosh, I watched so much CSI.
That's what I wanna do too.
Now I have students now who are like in a master's program for forensics. I'm like, I'm living vicariously through you, you know. But yeah, that was not for me. So I didn't particularly enjoy the research experiences I had. Like I was dehydrating alcohols with acids and using UV-vis spectrometry to measure the absorbance. Then like the Guggenheim mathematical method to characterize, what the kinetics were of the reaction. And it's strange because the actual topic I did not like. But the whole experience of doing research, like having independence, trying to solve problems, I'm like, this is really, really cool. And so I actually ended up in grad school not because this is funny. Not because I knew what I was doing, but because I didn't know what I was doing. I had no idea what I wanted to do after undergrad. Like I knew it wasn't med school because I had shadowed in a hospital and like, it was cold and all the walls were white and like everybody was sick and I'm an extreme empath and I can't. Like I would just be sad all the time. And so my mom you know how moms talk about their friends or talk about their kids to their friends? She's talking to her friend and was like, "Oh, yeah, my daughter, she's graduating and doing nothing with her life." And she's like, "Well, I have a friend at Loma Linda University and he does research, maybe he could talk to her." So I talked to him on the phone and he was like, "Hey, what do you think of research as a career?" I'm like, "What do you mean?" Like, I didn't know what a scientist was. I didn't know research could be a career. I didn't know there was a way to, like, build knowledge and actually contribute to what we have in our textbooks. So he explained that to me over the phone, and I was like, "Oh, yeah, that sounds really, really exciting." He's like, "Okay, well, apply to this program, take the GRE, and yeah, make it happen." So then that's what I did. I applied to one school, and then I got in, and really it was a learning curve. I was prepared, but I didn't know I was prepared. I was extremely naive about how grad school worked and what it was, but I joined the IMSD program. So it's an NIH-funded program, Initiative for Maximizing Student Development. And basically they taught you like this is how you be a scientist. This is what it means to like create knowledge. And so my major, I guess, applied to the biochemistry track because that's what I knew as an undergrad. But when I was rotating to find a lab, I found an immunology lab. And once I got a taste of the immune system and how it works, that was literally my first exposure to learning immunology. I had no immunology classes in undergrad. I had very little bit. I had a very small amount of bio background because, like I said, I was a biochem major. But I fell in love with it, and I couldn't, I couldn't decide to do anything else. Like I couldn't I don't know, the immune system is just so beautiful, and I don't know how else to explain it. And I have a mentor who says
I kinda like
th
it too. I made a
yeah.
podcast about it, so
Right, right, exactly, yeah. I have a mentor who's like, "Something's wrong with the body, we don't know, like, how it's working, it's the immune system," like, that's always the answer, and I kind of approach the world through that lens of, like, the immune system can touch every single system, every single organ, it has access to basically every place in our body. So, you can use it to heal, to protect, and, even to hurt, so understanding, like, how the immune system is intersecting with all these different fields that historically have been very, very siloed, I think it's extremely powerful.
I agree. And I think your background in chemistry kind of gives you a little bit of a superpower too, because you bring that outside perspective and maybe a better ability to create new assays and troubleshoot and be more of an engineer in the lab, which sometimes I feel like I'm always lacking. I look towards my mentors who are much older than I am, and they have such good biochemistry knowledge. And, I use all these kits. They know how all the kits work, and I'm like, I didn't get that training. So I think having the chemistry can really help you. And any really multidisciplinary training can really help you later on. You went on to publish, after your PhD, a very high impact paper in your postdoc at Boston. And we touched on this a little bit. That was in the lab of Barbara. I'm going to butcher this. Is it Nikolakzik? Lak-
Nikolaichik.
Nikolajczyk.
Yeah,
Okay.
yeah, Imma send her after you. Like, I mispronounced her name once and then, like, never again.
And then you came back to California for a second postdoc, as you mentioned, with Pamela Mellon and Mark Lawson at UC San Diego. And you mentioned you did that because your first PI from the first postdoc was gonna move, and so you would needed to find a new lab. But you'd already published this big paper. Maybe I'm, getting the chronology wrong, but I feel like you already would have been a great candidate for an independent position after the first postdoc. So I was wondering, why did you choose to pursue the second postdoc?
Well, part of it is that I had a vision for the question I needed to answer, and up until that point, I have worked in all human-only systems. So, human cells from blood donors, human cells people with type 2 diabetes, plasma sample, serum, and really I had never worked with animal models or being able to perturb things that happened over time. I always had like a screenshot and doing like comparing two different groups type of thing. And I knew that for the questions that I wanted to answer in my independent laboratory, I needed to know how to work with animal models. And so really that was the experience that I was looking for before I wanted to venture off and say, "Hey, I'm ready to be independent and ready to launch my own lab."
Did you have people telling you that you could go start your own lab now, but you were like, "No, I'm not ready. I need to get more training first"? Or did you just not have people telling you that? Because if people were telling you that, that might be hard to resist.
I mean, I had one person who was like, "Dequina, you can apply. I think you're ready." And I'm like, "No, no, 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 no." Sometimes, for better or for worse, like, I'll hold my cards a little too close to my chest. So it's like I didn't, tell my postdoc mentor, it's like, "I'm ready or not ready," or, "This is, like, the long-term plan." In the moment, it was like, "This is the research plan. Let's get the research done." It wasn't so much, like, career planning. And a lot of that I kind of did on the side myself. I always make, like, these five-year plans, five-year goals. Then in my second postdoc, I was part of another training program, which basically showed us how to use individual development plans with our PIs to do this long-term planning. So instead of me doing the planning by myself, I was doing the planning with my PI. And then it made it clear, like, okay, yeah, I'll be ready after these couple of things. And this is what will make me the most competitive, not just the most competitive applicant, but a competitive applicant that can land where I actually want to land. I don't want to just end up somewhere. I want to end up where I have the resources that will make the research vision that I have successful.
And you did that, right? You ended up in a very great place, UC Irvine. I would be thrilled to have a job there. That'd be awesome. So congratulations on
Oh, thank you. It's awesome here.
given your high level of success and your relatively recent transition to an independent faculty position, you probably have a lot of good advice about how to successfully transition to independence in academia. Could you share some of that advice? Maybe stuff that you give more commonly to most people? And I'm also particularly interested to know if there is any advice that you hear given to trainees often that you think is bad advice.
Ooh, okay. So let me start off with once you secure a position, the importance of actually planning and knowing what your vision is. A lot of times we're just grateful to like, okay, we got the opportunity. And then everything turns into I'm problem solving when something kind of hits me in my face instead of trying to think of what are the problems that I'm going to run into and then how do I prepare for that. So any space and time you have to plan, you can plan. Like say even before you're ready for the job market, like sit down with yourself, and this is like part of putting together your faculty application packet, right? What is my vision for the lab? What do I want to accomplish? What do I want the environment in my lab to be like? How do I want my PhD students to be trained? Like, what type of PhD students do you want? What type of personnel? Like, for example, I know very clearly, like, I need students who are self motivated, care about the overall mission of the laboratory, innately curious, because I'm not a micromanager. I'm like a cheerleader, like woo woo. And so, if you don't have something for me to cheer on, it's going to be hard for us to have a mentorship relationship. Like, I also know that I'm probably like TMI, and when I interview students, I'm like, "Look, in this lab, we know about each other's personal lives. If you're uncomfortable with that, you will be very uncomfortable in this laboratory." So being clear on, like, how you work as a mentor and thinking about your mentorship philosophy is gonna help you recruit people that will also help you be successful, and thinking about how you can help them be successful. So a lot of that forward planning. There's actually, a paper that me Tina Termini actually coordinated, like, writing this and putting it up about how to transition once you have a faculty position, things you can request ahead of time. Like, sometimes you can literally get access to, say, all the online trainings before you start. You don't have to wait till you get there. Things that take a long time to order, you can ask, "Do I have access to ordering systems? Can I go ahead and get the renovations done before I actually start? Can I order in the big equipment so that I'm not just sitting here waiting for equipment to come in?" Or if you're trying to recruit, like, once you know you're going to have a position and have money, like, start advertising and looking for people, right? And the other thing I emphasize is using your network and senior faculty for those first hires. A really bad first hire can sink your lab very quickly. It takes a lot of energy and effort to have someone in the laboratory who either isn't motivated, isn't doing well, brings the whole environment down, and then people don't want to be there. You want to make sure that your first hire is a good hire. So don't rush into just having a warm body in the lab. Like, don't just be, "Oh, I just need a person, I need a person." Like, no, make sure it's the right person. Okay, so you also asked about if I hear bad advice
Yeah.
being given. That is a really, really good question. Huh. I mean, I hear like conflicting advice more so than bad advice.
Or advice that you don't agree with maybe?
Yeah, I mean, okay, I know there's always advice against too many undergrads or growing too soon, like growing a lab too large too soon. And I think part of that is you knowing what your capacity is to mentor. I also hear advice that, you should try to move away from the bench sooner rather than later. I also kind of disagree with that, and especially when your lab is young, I think you need to be very, very involved with your trainees until you fully trust everyone to train other people. I've seen this happen with some colleagues where, like, you hire people, you assume they know what they're doing, you step away, and then you have a huge mess on your hands. And so being really like, you don't have to be overbearing, but being hands-on at the very beginning, especially with your first couple of trainees, I think is really, really, important. And yes, you do have to divide your time because you have to focus on, grants and things. Oh my gosh, that reminds me. Okay, so this is some advice that I think I push back on a lot. So you have this almost fight of people saying like, should you focus on grants or focus on papers? Focus on grants or focus on papers? And they almost make it seem like you have to pick one or the other, and then in all honesty, like, you have to do both at the same time. When you first start, there is gonna be what I call some dead period and, like, a lag. Use that time to write ferociously and get drafts of things together. Like, when you cannot physically do experiments in your laboratory, you should be putting out grants. And at the very beginning of your career, you have your strong postdoc publications, and people will fund you off of, like, promise and potential and hope. But then as soon as you hit about year 1.5 to 2 years, it's basically the doldrums, and you're not gonna get funded until you've shown that your lab can be independently productive. So I almost call it like the bait and switch. Like grant, 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 grant. When you start at the very beginning, use every resource, all the ideas that you have, all the preliminary data that you brought over from your postdoc, and then get your lab up and running. And I say get to first paper, whether it's small or big or whatever, have something published with your lab as senior author by year 3. Like that's really the timeline you are shooting for a project. It doesn't have to be your big shiny because good science takes a lot of time. Those really high impact papers, they take a long time, and that's okay, and that's fine. Just know that you have to move along smaller projects first so you can give funding agencies the confidence that you can execute as an independent lab.
That's a good one. I think I know another piece of advice that you may think is bad. A lot of times you hear people say early on in your career, you should focus on one research direction. Your lab has at least two. I feel
Mhm.
like you would disagree with the focus your research direction to one. Do you disagree with that?
I partially disagree. I understand why, and once again, I think this comes back to knowing your own capacity. But what I love about having 2 clear research directions, they are interrelated, but they're also completely separate. It means that I can try to fund the lab in completely different places, right? It helps me diversify my funding portfolio, and also it's fun to hop back and forth. And then techniques that can apply to one field, we can adapt to the other field, and the students learn from each other. So for the most part, what we have is like half the lab is either in type 2 diabetes or they're in PCOS. And then when we have lab meeting, each are learning about the other and then applying to the other's projects, or they're helping out or learning new techniques. And then some stuff that we never thought about in diabetes, say like some sex differences, or like, oh my goodness, like what's happening with the ovaries and the CD1A mice. There's all these things happening that we wouldn't have never looked for if the students who are working on PCOS like weren't involved with the type 2 diabetes project. So they kind of cross pollinate. And my advice is for the most part to follow that, to have like one clear focus as a junior faculty, because it is very difficult to manage 2 lines of research. Like I have to have turn on one brain and then switch the other one off and then vice versa. But once I had the vision for my lab, it was very clear to me I couldn't pursue one without the other. They're both deeply important to me and questions that I needed to address, and I felt like one could not wait. So I had to put in extra work to make both of them actually happen, and in order to move 2 different lines of research, you need a big enough lab group, so I had to recruit more people than I had anticipated so that, the research would move forward. But that's actually, I think, for the most part, sound advice from senior faculty, where they say, like, focus on one clear question, one clear direction, and it'll make it easier for you to be productive, and then also to get funding once you produced and published.
Dequina, you are quite different than most people in academia. Your lab website has a tab titled Why We Slay, which I can confidently say I have never seen on any other lab website. You are super confident and so authentically yourself. How has the mentorship you have received influenced how you show up in academia?
Oh my gosh. I love this question. I spent a lot of grad school honestly losing myself, losing my authenticity. So I have always loved pink. Pink is like the most beautiful color in the whole world. It's bright and happy. And then a close second behind that is yellow. It's the color of the sun. It's like it's magic. And so I used to have my closet just organized by these bright colors and bright colored clothes. And I went to my first research conference and I was like, oh, I'm not supposed to wear these colors. So then I went through a couple of years of actively removing color from my wardrobe. And I felt like I was getting drained of like my life and everything I loved as I was trying to adapt to what I believed a scientist is supposed to look like. Like it didn't feel good. It wasn't comfortable. Even the fact that like say I put on an outfit that I thought was cute and then I'd hear like professors later say like, "Oh, that was inappropriate what she wore." I'm like, "Oh my goodness, I really got to figure out what to wear to these professional spaces." And then my postdoc mentor, Barb Nikolajczyk, the first time I met her, like, it was life-changing. Picked her from the airport, she was giving a talk at our school, and she's like, "Oh, yeah, I'm gonna go to the Grand Canyon and ride horses and donkeys, whatever, and then hang out with my son." I'm like, "Oh my gosh, she has a family, she does fun stuff, she has a life," and I read her papers are so good, it's amazing science. I was like, "I want to be like her. I have to be able to work with her." So when I got the opportunity to work with her, like, that same woman I met that day was the same woman that is like big boss lady, highly confident. Like she is who she is, and no one can tell her anything different. The first week I worked in her lab, we went on a paddleboarding adventure. She's like, "Yeah, look at this lady just out here paddleboarding. This is great. Yeah, science." And, like, she would joke and laugh. I remember one time I walked into her office and she was, like, playing Lemonade, like, this beautiful album, in her office, like, while she's writing a grant. I'm like, "Wow." Like, she is unapologetically herself, and she's done this and has a family and is successful. You know what? I can have my cake and eat it too. She was my first example that you could be yourself and still be successful in science. And so after working with her, I decided I'm tired of the exhaustion of trying to conform to what I think they want me to be. It's like, I'm gonna be the best scientist I can possibly be, and then they can take it or leave it. And then so, reverse course, right? I started putting the color back into my wardrobe over time while doing my second postdoc in San Diego and just being myself and having a good time and having fun doing science. And it was so much easier to enjoy the struggle of problem solving, but I didn't also have to have the weight of trying to conform to like what was professional around me. And so I decided, like, you know what, this is how I want to navigate through the world. And then now that I have my own lab, I want to empower all my students to bring their whole authentic selves to lab. Like, if you wanna be goth and wear all black and do your science, do that. Like, if you wanna dress up some days and, like, have your nails done, let's get some good gloves that won't break when you put your nails on so that you can, like, do your science and, like, still be you and what makes you, like, feel good about yourself. And so I'm really into confidence building, accepting yourself as you are, like, the way you were created, and to do the best science. And I think when you do that, you're able to come with clarity to problem solving and bring, like, your own perspective that I think is very, very powerful. Like, when I say the students in my lab are so creative, like, I'm not just saying that because I love them and I'm biased. Like, no, they have brought approaches to the questions that I have that I would have never thought of myself. They feel empowered to be like, "You know what? I have an idea. Let's just, like, shoot my shot and see how it goes." And then we do that, and we uncover, like, novel pathways. Like, this whole, the macrophages are regulating hormone thing, like, I wouldn't have found that. I was over here looking for T cells in the in the pituitary just because I study T cells. And so I love this whole idea of, like, just being my authentic self. Like, not everybody likes it. You're gonna have haters, like, I'm fine with that. But the people who see that I am genuine and I mean what I say and I say what I mean, they can get along for the ride and we can build something beautiful together. We can truly move science forward by using our different perspectives.
And I think being authentic, I'm sure, helps with imposter syndrome, which you hear about as a very common thing in academia, people never feeling like they're supposed to be there. But I feel like if you are just yourself, it might sort of vaccinate you against the imposter syndrome.
Yeah, it helps. Like, I like the word vaccinate against imposter syndrome, right?
I had to
Because
throw that in there.
because you're not immune from it, right? So the vaccine doesn't stop the infection, but it makes you fight it off a lot better, right? And so I'm even just last weekend, I'm like, you know what? What if my resilience is gone? Like, what if I cannot bounce back from this? We had a paper get rejected and it was a gut punch. Like when I say this paper, like it's so good. And this is the first study showing that we have direct cause and effect of the immune system in PCOS. And this was a Herculean effort between like my lab and then a genetics lab of my friend Marcus Selden. And like so getting that rejection, I'm like, I just don't understand. It's like, no, maybe I wasn't built for this. Maybe I'm not supposed to be here. Then like I sleep on it, I'm like, you know what? No. Just because one editor thinks it is important doesn't mean it's not important. And I was made for this. It's like I'm good at my job. I'm good at science. I ask good questions. I do rigorous work. It is replicable, right? And so we will get this published and people are going to see, and this will be the foundation of being able to have immunotherapy for PCOS. Like this is the beginning. And then so I'm able to pick myself up a lot faster than I have been able to in the past.
That's really good and super important. How have you, in turn, approached building the culture in your lab and mentoring your trainees after how your experience with this mentor that really allowed you to feel like authentically yourself? You mentioned a little bit about hiring, and you're pretty transparent about how you show up. And could you elaborate a little bit more on how you're building the culture in your lab?
Yeah, so I started off by leading by example, and I think that's the clearest way. So for me, like I said, when I saw my mentor, I was like, "Oh, she can be herself and she's still respected. You know what? I can do the same thing." So I actually, well, I don't like being by myself. So I sit in the lab every single day. And so they're used to just kind of seeing me around. And when we have conversations, one of the things I think is very important is to acknowledge like where I've made mistakes and where I've messed up. So this comes transparency. Like transparency, I think is key for them to actually trust me and trust I have their best interests at heart. So making sure that I show up for them, that I'm putting in effort. They see like how many hours I work. They see when I come in and they know when I leave. They have some access to my calendar so they know like when I'm busy, when I have meetings. When they come to their one-on-one meeting and then they messed up on something, it's not like, "Oh, I can't believe you messed up." It's like, "Okay, now how do we problem solve this and how do we learn from this? How do we grow from it?" And they get comfortable with what I call productive failure. And that's really a key tenet of like how I train and how I want people to be transparent when they're training others. So I failed at things. This is what I learned from it. This is how I got better. Now you failed at this. Let's learn from it and get better. And then it just keeps on building. So I've actually overheard my students talking to their undergrads being like, "Oh no, no, it's okay to fail. We all make mistakes and mess up. This is when I made a mistake." So they're literally doing the same thing that I have modeled for them for the undergrads. So that's kind of my approach is as much as possible to model the type of environment that I want them to have in the laboratory. And this even extends to like caring about each other on a personal level. When I think about like networking and collaborating, it's literally helping a friend. So if you have someone that you care about, that you are friends with, when they're in trouble or they're in need, it's very easy to be like, "Oh, let me help you." When it's just a colleague or someone you don't know, then it's like, "Oh, now this is extra work on my plate." So my goal is also to build these genuine friendships and relationships within the laboratories, like they're close colleagues and friends to whatever extent that they want to be. And so, for example, like when it's a birthday, I will bake a cake. Like you tell me what you want, I'll bake whatever you want, and we'll see how it goes. So I generally show them like, look, I love you and I care about you. If we have something to celebrate, we make sure like we all celebrate. I do little like lab gifts and swag and make sure that they know was like you are my priority. When we have our one-on-one meetings, even part of our meeting that's like, how's your mental health and your wellness? Are you taking care of yourself? What is the plan, not just for the research, but for your career so they know that I'm invested in them? And it makes it easier for them to trust me and then to invest in others. And they see that this is a environment where we are supposed to support each other. And so that's my approach.
Can I join? Sounds great. Speaking of failure, I like the topic of, or the phrase you said, productive failure. I've looked at your CV, it's on your website, and it's incredibly impressive. Long list of papers, talks, awards, successful mentees, a company, the list goes on. You also had a baby the same year you started your lab, I believe. Someone looking at that CV may think, man, she's batting 1,000, always hitting grand slams. Can you think of a time when you failed at something in a big way? Like you said, I think it's important to remind people that no one is perfect and that as catastrophic a failure may seem at the time, we can get past it and do great things. And sometimes great things actually come from the catastrophic failure.
Yep, I have one that popped right into my head right away. Okay, 2018. I've had from then, like 4 years of like postdoc experience, have a big paper coming out. I went on the job market. I applied to like 20 institutions. I didn't get one interview, not one. While the same people in my same training program and the same cohort, like I literally would take my CV and look at theirs and be like, I don't understand. Like I'm just as good as them. Like, was I not made for this? Was I not supposed to be faculty? Maybe I'm just not good enough. And all this time, all these years, I've been lying to myself that I could actually do this. And I found myself in such a depression because when I say that I wanted to be faculty so bad, like this is plan A. There's no plan B. There is no backup plan. There's no, oh, this is just it's just a job. Like that is not my perspective. Like for me, being faculty is my goal in life. It's my mission. It's my ministry. It's what I was born for. So then having to run into the idea that maybe this is not going to happen for me and that I wasn't good enough and I wasn't cut out, like that cut and that hurt deeply. And I had a friend who sat with me, and what's funny is because like, she's somebody we both applied to the same position, and she got an interview and actually got the position. But she was like, "Dequina, your time will come, and I want you to relax. It will be okay." Like, she's just a very, very calm person, and there's something about her just kind of saying that without, like, the panic. Because 'cause I'm a very, like I go extreme on whatever it is. If I'm happy, it's really extreme. If I'm sad, it's super extreme. And I was like, "You know what? Maybe she's right. I'm gonna step back, talk to my PI, and look at my application and say, how can I make this better? What can I do in the next years?" So in that following year, I published another paper. I ended up getting a K99 R00. I had some co-authorships going on. I went to every single job workshop thing there was on campus, got feedback on all of my materials, and I was like, you know what, I'm going to try this again. And when I say I spent a lot of time personalizing every single application to where I was going to apply. I spent a lot of time. And so the second time around, I applied to less positions, but they were so like targeted and specialized. I got advice on how to actually like contact the search chair or see if there's somebody that I know to talk to about the position to get more information and show my interest. And then the second time around, I actually got multiple offers and I had opportunity to choose where I wanted to land. So it was such a big night and day between like this is never going to happen for me and like, oh my gosh, now I am living my dream. Like I get to mentor students and teach and help people achieve their own dreams. And I couldn't imagine doing anything else. So yeah, that was one of the biggest fails that I had. And I decided I wasn't going to give up. I was going to try again.
There's hope.
Yeah, yeah, there's hope. There's hope.
In an interview with Endocrine News, you said, "I want to win a Nobel Prize."
Hell yeah.
This is something I presume many scientists may think about occasionally, but few make it a goal, and fewer are bold enough to say it out loud.
I know, right? I sound like jinxing myself.
What drove you to make winning a Nobel Prize a goal and to say it in that interview?
Okay. So actually, personal things drove me to say that. So I'm going to step back a little bit to my experience as a grad student and as like a Black woman in science. And there's so many spaces that I've been in since graduating undergrad where I felt like I didn't belong or I wasn't welcome or I wasn't wanted. Even going all the way up to like looking for faculty positions. Like you go to a department and you go through and you scan everyone in the department and you get this weird sinking feeling like, I don't think they want me there or would like me there. It does not feel good. I literally like when I I shouldn't put him out there like that. But like when I met my husband, my friend told him that, "Oh, yeah, she's in grad school." He's like, "Oh, what? That girl's in grad school?" Like, he was surprised I was doing a PhD. Like, I did not like that feeling. Like, why are you surprised that I can be doing science, that I'm smart? Or even I had one interview that I went to, and I met someone in a coffee shop, and I told him, like, "Oh, yeah, I'm on a faculty interview." It's like, "Oh, yeah, so what department are you doing social sciences?" Like, the assumption that a Black woman can't actually do science. I'm like, not cool." And then so I recognize the only way to change public perception is that the public has to see something different. And the most public display of being good at science is a Nobel Prize. Like, I want people, when they imagine a scientist, to have a whole bunch of different people come to their minds, not just like the white guys we see in our textbook, because it's very, very like mono. And at the end of the day, like in my class, I do this. We have a lot of Nobel Prizes where there were teams who contributed. And then not everyone, especially the minorities and the women, they were not like congratulated or given their flowers for the work that they did as part of that major discovery. And so I don't want that to be the case anymore. So the reason why I decided to go on record and say I want to win a Nobel Prize is because winning a Nobel Prize puts you on the world stage as being the top of your field and having done something that really contributes to society as a whole and moves human health and science forward. And to see that a black woman and no black person has ever won a Nobel Prize in the sciences, like period. Like it was really, really sad. To show them that like we are good scientists too. Please don't make assumptions based off of, like, what someone looks like, and I want it to be that anybody who wants to do science can do science and is respected for the work that they do and the science that they do, how they approach questions, how they problem solve, and not necessarily where you think they come from or what their background was. So, yeah, that's why.
I truly believe that having a strong why is critical for having the motivation that it takes to get through the hard times. And I think you have some really strong, wise reasons behind why you do what you do. You know, what you said just now, and then what you mentioned before about type 2 diabetes running in your family. And it sounds personal. And based on what you've told me, I think you can do it. So you got someone rooting for you. I previously alluded to a company that you started. It's called First Gen in STEM. Why did you start this company and what's it all about? What's the mission?
Oh, okay. So let me start off. I very much believe in like diversity, equity, and inclusion. And given kind of how we see DEI in America, like I saw it bubbling up, right? I grew up in Florida. I've kind of been in like different pockets of spaces, people with like different political backgrounds and ideologies. I think at the end of the day, everybody wants access to be fair, and how we get there differs. And what I've seen is that people may feel like there's discrimination or not. And I wanted to create a program that really like rallied around our shared similarities. So, my experience being like a first generation person in the United States trying to navigate higher education, it was basically like I had no idea what I was doing. And I was very blessed to have mentors and faculty like look at me, see my potential, and be like, I'm going to help her get to the next step. But not everybody runs into these random mentors and faculty that are going to help them. And everyone knows what questions they should ask. Like they just don't know. So, whether you're like a first generation immigrant, whether you're the first in your family to go to college or like do higher education, whether you're from like a poor rural family that doesn't have R1 universities that have all these access to science and things like that, we have this shared commonality that we don't know how to navigate higher education. So the idea was that if I could put together resources that are centralized that can reach students who don't have basically the human capital to figure this out and the resources, that it could be freely accessible to them. There are a lot of really, really good programs that some are funded by the NIH, some are funded by private foundations that essentially look for students from historically excluded groups. So this would be like maybe African Americans or like the Latinx community or like Native Americans, indigenous, or like even those who come from low socioeconomic background, disabilities, and people will build these programs to say, how do we build community? How do we build support? How we get them the tools that they wouldn't otherwise have access to because historically they have been excluded from access to science. But what all of these different groups have in common is that they don't know how to navigate it. So it's like, how can I bring access to everybody together? So the mission of First Gen in STEM is to build a repository of first stories of people who are also first generation who are able to navigate it, because there's something about seeing someone who's similar to you who's been able to do it that gives you the confidence and tells you I can do this too. So that's number 1. Number 2 is to provide resources that are freely accessible. So for all the students who can't get into like these cohort-based programs, there's just limited space, that they can still get the same information that the students have access to. So for example, the very first resource that we're building, we should have it up online soon, is how do you choose a graduate lab and manage rotations? Like so many students, I've actually seen some students here. You have some students whose parents were academics and they're like, oh yeah, I got to set up my rotations before I even get to campus. And you have students who get here, they're like, oh, I was supposed to set up rotations, right? It's very, very different. And then all their fall rotations are already taken because they had no clue. And so the idea is to make an even playing field by giving knowledge to everyone. So that's really like the two-part mission is One, to show people that you can do it despite your background, like where you came from doesn't dictate where you go. And then number two, like here are the tools that will make you successful.
Since Trump was put into office for his second term, people have been saying that DEI is dead. What's your reaction to that as someone who's, a DEI advocate?
My first reaction was actually intense pain. It was very, very hurtful because, in all honesty, I am the product of DEI. I am someone who was highly motivated. I was basically valedictorian of my class, top of my class in undergrad. I finished undergrad in 3 years doing advanced studies while also being an acrobat while doing extracurriculars. Like, I worked hard. I worked my butt off. I just didn't have direction and I didn't have a network to know what to do. And then so these programs allowed me to understand like how the system works, how to navigate higher education so that I could become the scientist that I am. Like we are so close to understanding and figuring out the root causes of type 2 diabetes. And without DEI, like none of this perspective would have been possible. Like I get to be this cool interdisciplinary science because of DEI. And so I look amongst the different faculty and what people care about, and those who understand what the actual core values of DEI are, what like the core values, so it's diversity, equity, inclusion. And really with equity and inclusion being at the forefront of that. We want everyone. Like when I say everyone, I mean everyone, even the people who say they feel excluded. The whole point is that nobody gets excluded, that everybody has a fair chance to access higher education. And so I don't think it's dead. I think it's one of those things that those who have a spine and who care about the true value system of DEI, they're going to fight for it. And in whatever way they can fight for it, they will. Like for example, right now I have a quote unquote DEI supplement to my grant studying diabetes. What the supplement actually pays for is for us to look at the immune compartment in the pancreas in humans to figure out what type of inflammation is causing the islets or the beta cells not to work. And then as part of this, because I care about mentoring my students, I took on 3 undergrads who were transfer students, so from community college. And at community colleges, they don't have access to research labs, and they get a chance to be a part of the research enterprise and to gain mentorship and for me to help them in that transition. So this project is exactly what these undergrads are going to work on, and we call it the DREAM program. And so, like, that's what DEI funding is supporting. And it's not that these students aren't qualified. It's not that these students didn't work hard. It's not that these students don't have amazing grades. Is that they started off with less resources. They went to community college. Once they transferred, it's very difficult to join a lab when you're a junior or senior because faculty are like, if I invest in a junior or senior, they're going to get trained and then they're going to leave. So instead, I'm trying to create a mechanism where we can help to support these students who transfer and they don't have the same fair chance at getting into a laboratory that other students who enrolled as freshmen at this university did. So that's really what DEI is. It's how do we let the students who otherwise wouldn't have had an opportunity get the same opportunity that others have?
So do you think that DEI wasn't really being done appropriately and now it needs to be corrected? And
I think so. I think so. So after George Floyd, when everyone was like marching in the streets and stuff got crazy, I think DEI became kind of like this buzzword and thing that was cool. And so a lot of companies and universities implemented all these programs because they felt like they had to. And I think they weren't well thought through. And I think all it takes is a couple of almost like abuse of power to sour someone on a whole entire ideal. It's like DEI, quote unquote, has been like happening for a long, long, long time. It's basically just helping underserved communities. That's what DEI is. But once you have this whole like politicization of like DEI, which happened like in 2020, then people think of it in a way that it shouldn't be thought of. And then there are places where like it's not applied appropriately. So I can give you a really clear example of how it's supposed to be applied. And I can give you an example of, maybe a way it wasn't applied correctly. So say we are on a search committee and we're searching for like a new faculty. A lot of times what will happen is we'll make the job ad and then some senior member will be like, "Oh, I know somebody perfect for this. Let me call my friend and tell them they should apply." Not okay. Our DEI office or our equity advisor, they say, "Hey, how are you going to advertise the position? Where are you going to advertise? Are you advertising in a lot of different places so you can get a very diverse pool of applicants?" That's a DEI principle, as opposed to let me call my friends and just tell only them to apply. So that's how it's supposed to be applied. And the idea is to invite as many qualified, diverse people as possible. And then so what people's perception are is they think that for some reason applying DEI is I'm going to pick someone who's underqualified and choose them over someone who is a white male. And that is not what is happening. And if it was, we'd actually have a lot more minorities in positions than we do, as we're still underrepresented. And we're working very, very hard to basically acknowledge for the work that we do. But yeah, it's one of those things where like I don't think DEI is dead. I do understand that there is clear concern for discrimination, and at the end of the day, like I don't want anyone to be discriminated against. Like the whole goal, as I said, with DEI is supposed to be inclusion and that everybody has equal opportunity. So any way to make that happen, like I'm okay with. But my heart does hurt seeing that people are like even myself, like I've had people write me on social media or in my DMs and say, "How many white people had to be passed over for you to get your job?" I'm like, "Well, excuse me, have you seen my CV?" That's like,
It's long and very impressive.
I yeah, so people are starting to have this automatic thought process that like if it's a minority in a good position that we were chosen for DEI. And what that does for a lot of people is actually it attacks their sense of self-worth. Like, "Oh, no, maybe I am DEI, maybe I didn't earn this." I'm like, "No, like, personally, I know I earned my position. I worked extremely hard." Like, I had to go through being rejected for a whole, application cycle. I was like, I know I'm not getting chosen just because of skin color. I'm getting chosen because I was the best scientist for the job, and I'm showing that here with my lab now."
Yeah, I guess the hope is that DEI can give everyone a chance,
Yes,
and then the best candidate will still rise to the top,
exactly.
which sounds totally reasonable.
Yeah, yeah, exactly. So if we can get it to always be applied that way and to help with public perception of it, I think there's hope for it.
Getting pretty close to the end here, so I just wanted to ask before we wrap up, is there anything that you wanna share with the audience that we didn't have a chance to touch on today? Anything at all come to mind?
Oh, yeah. Okay. So I'm a scientist. I run a lab. I do research. We're trying to advance knowledge, but there's actually another part of my job that takes up a lot of time and that I love very, very much. So, as part of my position, I also teach the undergraduate classes, the students that come here for a degree and want to go to medical school or go to pharmacy school or be an optometrist. And I get to teach them all the basic science that they have to know in order to do well in those professional schools. So, part of supporting scientists like us is also supporting the education of the future generation of all of our health professionals. And I truly see this as a privilege and an honor. And I know currently with that, the whole political climate, everyone sees like scientists are the enemy and we're grifters. I was like, I sad that that is what you see, but we're also here to support training of the future generation of your children, your kids, they work very hard to get these degrees. We wanna make sure that they have the knowledge that they need to be the health professionals that will support us. Like, the same way that policemen are service members, the same way that teachers are service members, scientists are also service members too, and we are here to serve human health.
Awesome. Thank you so much for your time, Dequina. Where can listeners go if they wanna learn more about you and your research? I know you're very active online, so could you share, like, your different handles for things?
Oh, yeah, we're actually in a lot of places. So you can find us on TikTok, that's Nicholas Lab for the handle. Instagram, that's also Nicholas Lab. Me personally, I'm on Blue Sky, so at Quina Science, Quina Science. And also you can find me on Twitter/X. It's a little crazy there, but I'm there. And I think that's I'm on LinkedIn. I don't really post on LinkedIn, but I'm there if you want to connect or like reach out. And then if you just want to know about the science, we try to keep the website updated. That's the nicholaslab.uci.edu. All
Thank you so much, Dequina. It's been great.
right, awesome. Thank you for having me.